Hello everyone. So this video is all about the basic structure of amino acids. What are amino acids? As we all know, proteins are polymers of amino acid. Each amino acid residue joins to its neighbor by a specific type of covalent bond. There are in total 20 different amino acids which are commonly found in proteins. First one was discovered in 1806 and it was asparagine. Okay, so the amino acids are alpha amino acids in nature instead of one, that is proline. क्यों? क्योंकि प्रोलीन में जो है अल्फा अमीनो रिंग नहीं होती है, उसमें इमिनो रिंग होती है. सो देयर इज अल्फा इमिनो रिंग प्रेजेंट इन द प्रोलीन, सो इट इज नोन एज द अल्फा इमिनो एसिड. ओके, amino acids have a carboxyl group and a amino group bonded to same carbon atom, that is alpha carbon, differ from each other in their side chains or R groups, which vary in structure, size and electric charge. This overall affects the solubility of amino acid in water. As you can see in this picture, this is the amine group, this is carboxyl group, this is R group and this is hydrogen atom, all bonded to a single carbon, that is alpha carbon, right? So alpha carbon atom is a chiral center. Why? Because of tetrahedral arrangement of bonding orbitals around alpha carbon atom, four different groups can occupy two different spatial arrangements, and thus amino acids have two possible stereoisomers. Since they are non-superimposable mirror images, so they are known as enantiomers. We all know about the enantiomers, that these are the non-superimposable mirror images of each other. All molecules with chiral center are optically active. So there are in total approximately more than 900 amino acids are present in nature, out of which 35 are proteinogenic. For example, hydroxyproline, which is present in collagen. Okay, we can also take the example of gamma aminobutyric acid, T3, T4 amino acid, citrulline, etc. Out of these, 22 are being coded by genetic code, and out of these 20 are coded by standard codons and 2 being coded by stop codons, that is selenocysteine and pyrrolysine. The most common form of amino acid found in nature is L form, although D form of amino acids are also present in nature but for a unique function. We can take the example of toxic venom, that is country fan. It is exclusively made up of D amino acids, right? So amino acids are represented as one word citation and 3-word letter code, as you can see in this picture or the table. Many amino acid has one letter code which is not initial alphabet of their name. We can take the example of, for example, we can take the example of phenylalanine. You can see the 3-letter code is PHE while the 1-letter code is F. This is due to the unique names provided to these amino acids according to their history, according to their evolvement, right? So now moving on to classification of amino acids. Amino acids are classified on the 3 bases. The first one is nutritional value. Here we can see the nutritional value. Under nutritional value, essential and non-essential amino acids are present. Essential amino acids are absolutely required in our diet, while the non-essential amino acids are present in our body. Our body can make these amino acids if they are not present in our diet. Okay, in case of essential amino acids, we can take the example of glycine, alanine, tyrosine, right? While in case of non-essential amino acids, we can take the example of phenylalanine, tyrosine नहीं, threonine. यहाँ पे T है, थ्रियोनिन. जैसे मैंने अभी बताया था कि 1-word लेटर कोड डिफरेंट होता है कुछ अमीनो एसिड्स का, ऐसे यहाँ पे थ्रियोनिन के लिए हमने T यूज़ किया है और इधर हमने टायरोसिन के लिए Y यूज़ किया है. आई कैन शो यू अगेन. सी here it is threonine, it is T for threonine, and for tyrosine it is Y, right? You understand? So moving on to next classification, that is based on the metabolic fate. So based on metabolic fate, it is divided into 3 types: glucogenic, mixogenic and ketogenic. So what are glucogenic amino acids? These are the special class of amino acid which can metabolically converted into any of the glycolytic intermediates, major intermediate of gluconeogenesis pathway, and can ultimately be used to make glucose or its equivalent, right? We can take the example of all the 14 amino acids rather than lysine, leucine, and this Wi-Fi. Wi-Fi means, here I have I will discuss this later. First, let me come to the mixogenic amino acids, right? In case of mixogenic amino acids, these are the amino acids which are both glucogenic and ketogenic in nature. Lysine and leucine are exclusively mixogenic amino acids present in our nature. Right. Now, what is ketogenic amino acids? These are the amino acids degraded directly into acetyl coenzyme A, which is precursor of ketone bodies and different myelin sheaths. Right. The example is, I have write this Wi-Fi. Now again, if we go to that one word letter or three letter code, W is for tryptophan, Y is for tyrosine, F is for phenylalanine, and I is for isoleucine. Got it? So the last basis on which amino acids are divided, that is based on the side chain. That is, side chain is the R group. Okay. So yes, the aromatic amino acids or aromatic R groups. These are the groups, these are the amino acids which have aromatic ring in their structure. As you can see in this case, this is the aromatic ring, this is the aromatic ring, and this is the aromatic ring. Right. So these type of amino acids has aromatic rings in their structure. For example, phenylalanine, tyrosine, and tryptophan. Right. Non-polar aliphatic amino acids. These are the amino acids which are non-polar in nature, okay, which do not have a charge. As you can see, non-polar, you all know about the non-polar, meaning of non-polar. Okay, so this is glycine, alanine, proline, valine, leucine, isoleucine, methionine. Right, these are the non-polar aliphatic amino acids. Now, what is next one is the polar uncharged amino acids. In case of these polar uncharged amino acids, these are the polar one and having no charge. We take the example of serine, threonine, cysteine, asparagine, and glutamine here, right? And this coming towards the charged amino acids, we have in this category, first one is the positively charged R groups, in which the group side chains are having a positive charge, overall positive charge. Right, we can see the example of lysine, arginine, and histidine here. Here is having the positive charge group on the side chain itself. So these are the positively charged amino acids. And coming to the last one, that is negatively charged amino acids or negatively charged R groups. Here we can take the example of aspartate and glutamate, which both are having a overall negative charge in their side chains. Okay, so I will discuss the structure and detailed properties of all these amino acids in my next video. Also, this is my first video, so please comment down your views, suggestions, and also topics on which you want me to make videos. Right? Till then, take care. Thank you.